Welcome back. I hope everyone enjoyed lunch. I'm thrilled to introduce our next keynote speaker, Dr. Heidi Rehm. Dr. Rehm is the Chief Genomics Officer in the Department of Medicine at Massachusetts General Hospital and a Professor of Pathology at Harvard Medical School. She is an international leader in genomic medicine and human genetics, and she's been instrumental in charting the course for genetic variant interpretation. Welcome, Dr. Rehm. The floor is yours.
Thank you. It's a pleasure to be here. It's actually been a while since I've been back here at the HMS Conference Center, so this is great. So I'm going to talk about a few things around advancing genomic medicine, particularly through global collaboration and data sharing, which I spend a lot of my time advocating for. But for a few of the talks I've been giving more recently, I've been having some fun with the 10 bold predictions for 2030 that National Human Genome Research Institute put out, and a nice series of talks if you're interested in listening to leaders in the field give their views on each one of these. And I don't have time in the next 40 minutes to go into all of these, so I picked 4, and that will guide some of the comments I make in this space. So the first one was around the biological function of every human gene will be known by 2030. And for non-coding elements in the human genome, such knowledge will be the rule rather than the exception. So when we think about figuring out the function of every human gene, which of course is a biological endeavor, but it's also critical as we think about diagnosis, particularly of rare diseases. And one of the critical ways we understand human disease is actually to disrupt function of genes, and that can be through either observing the disruption in humans that have certain diseases or generating animal models. And this has been a very robust process we've used over the years to understand the function of human genes. So where are we right now with understanding both the function of genes and their role in disease? Well, if you use OMIM, which is the primary source of defining gene-disease relationships, at least those that have been claimed, we're at about 4,684 genes have been implicated in at least one disease. And in fact, many genes are actually implicated in multiple diseases. However, when we run routine analysis of patients with a suspected genetic disorder, on average, about 75% of those cases, we can't solve and identify the answer. So it tells us that we still have a long way to go in terms of understanding genes and their role in disease. And in fact, some of the projections that have been done looking at databases like gnomAD and others, and the mouse models and how they compare to human disease, there's evidence that there's probably over 10,000 more genes to be discovered for Mendelian disease alone. So to try to tackle that issue, Daniel MacArthur and I, now 7 years ago, I guess it was, started the Broad Center for Mendelian Genomics and the Rare Genomes Project to try to address this issue. For the Rare Genomes Project, it's been a direct recruitment of families across the US. So our IRB limits us to enrollment within the US. But our goal was to really help families find a diagnosis. And we, some individuals have had years of diagnostic odysseys, every possible test, can't find an answer. There are some that simply couldn't get access to genetic testing, didn't have the right insurance, or the insurance wouldn't cover, didn't have any insurance. So we've been analyzing these cases. We're now probably 1,000 cases in. And what you see in terms of, do I have a pointer here? Maybe not. I think so. So what you see is about 16%. We have, or 14%, there's a diagnosis with a known gene. So there's still, even when they've had everything, we end up finding, causes that are genes that are known. And some of that is the rapid evolution of our knowledge base. So if the family had testing 5 years ago and then we test them, it could just be we now know more than we did 5 years ago. In other times, they only had panel, and we need to do a genome or an exome. We're also finding novel causes of disease in these individuals, and we're able to work them up and identify and prove causality or collaborate with other groups to prove a causality over time. Now that's our direct enrollment arm. In terms of our Center for Mendelian Genomics, we've been able to broaden our reach by collaborating with amazing investigators around the world that have been recruiting their own families in different regions of the world in their own disease areas. And so we've amassed thousands of samples through this process, and in fact have 926 novel gene discoveries or candidates. About 322 of those are well-supported, publishable. Another 600 or so are candidates that are going through building of evidence. So it just shows you the power of collaboration, where we can enable a lot of science by working with some amazing groups around the world. I would say one of the most powerful ways that we implicate genes, in fact, almost every gene that we have implicated in disease over the last 7 years has benefited from what we call gene matching. And in this case, as we've continued the long tail of gene disease discovery, a lot of these genes are very rare causes of disease. And in order to find enough evidence to implicate those genes in disease, you have to find families that are in different corners of the world. And so about 7 years ago, we as a community came together to define the concept of gene matching and work to build a network where we connected databases around the world. And the principle here is very simple. You just put in your candidate gene. Maybe you've found a de novo variant in a trio, in a gene that's highly constrained, it's looking suspicious, but you have an N of 1. That's not enough to implicate a gene in disease. We all have about 100 de novo variants in our genomes, and so about 1 or 2 per coding region, and so that's not enough. But if you match it with another patient on the other side of the planet, has the same gene candidate and the same phenotype, and there's overlapping phenotype, that allows you to build evidence. So very simple principle, and in fact, we began building this federated network. It's probably one of the first examples of a truly federated genomic data sharing platform. What does that mean by federated? It means everybody's data stays in their own country, their own organization, their own locus. And we simply agree to query each other's databases. And it's like Go Fish. Hey, do you have a match for this gene? And you literally put it in and you wait on your email, and then bang, you get an email reply seconds later saying, you've got a match. It's really fun. So we've been doing this, and we now have, everybody in the rare disease space is using this database because it's so powerful. There's over 10,000 candidate genes in there from hundreds of thousands of individuals around the world. And we just published a special issue of Human Mutation talking about the last 7 years of use of this database. And I invited actually 3 clinical labs that have been using the database in the context of routine exome sequencing in their clinical laboratories. And those 3 labs alone have been part of implicating 1,000 new genes using this platform over the last 7 years. So just incredibly powerful platform based on opening your data set up to sharing broadly around the world. And this is just an example of one of the many cases that we've been involved in. Our Rare Genomes Project case is shown here on the left, small family, and we found 2 novel variants, one a truncating variant in a patient. We put it into Gene Matcher. We matched with the Gleason lab, and in fact became family number 8. They had actually already amassed through gene matching 7 other families. They also did functional studies, and so we lumped our case into theirs, and it's been published, and so on goes the story. And this is a common story that we have. So if we go back to that question, the biological function of every human gene will be known. Will it happen by 2030? My guess, and you can figure this out in 2030 if I'm right, we'll probably be at 80% is my assumption. I don't think we're going to quite be there, but we'll get much further than we are today. All right, so now I'm going to move on to the next one, which is generating and analyzing a complete human genome sequence will be routine for any research laboratory. And to be honest, I would argue that this is actually fairly routine already today, for the most part. However, not all genomes are created equal in terms of the quality of those genomes that are generated around the world. And the challenge is that there's constant innovation in sequencing technology, in variant detection methods, in the analytical approaches to identify causal variation, and the quality and completeness of a genome sequence that any one of us produces is actually quite variable given the constant evolution of all of these factors. And we put this table together for a recent paper from the Genome Medical Initiative to define all of the different types of variation, their diagnostic potential, and their maturity in exomes and genomes. And you'll see there's a lot of different variation that we have to look at in the genome, and they vary in terms of the maturity of detecting these variants. So these are challenges that our field is dealing with, making sure we have high quality analytical algorithms to detect structural variants, all the different types of structural variants, and so on. So I would argue that what we really need to do is work together to share all the tools. Every group is working on different algorithms and constantly evolving them, and it's hard to keep up with them all. But if we share all those algorithms together and allow everyone access to them, we will all have good, high-quality genomes to use in our patient diagnosis. So one of the ways that we're enabling this is through the Anvil platform, which is a federated cloud platform to manage, store, and share genomics and related data, as well as enable analysis and facilitate collaboration. And so you basically, everyone shares their data on the platform, and this is funded by NHGRI and all of the grants that NHGRI funds. They require their data to be put onto Anvil. But also then all of the computational scientists can put their tools and algorithms on this platform, and then your data's there, the algorithm's there, you can marry them together. And in fact, all of the data from the NIH-funded Centers for Mendelian Genomics, which is circled there on the left, has now all been deposited into Anvil, and so anybody can go and use that data and test out your new favorite algorithm to detect variation. We also have put our Seeker platform onto Anvil. This is a rare disease genomic analysis tool. It has lots of functions that I don't have time to show you, but on our website, our Seeker website, you can go there and watch little videos on how to use all the tools, the platform. So you can go create a Google bucket on the Anvil platform, load your data up, put it into this tool, and then be able to analyze it like anyone else would analyze a genome, looking for variation that might be causal. The software is open source, so we've been collaborating with groups around the world that are also using it, contributing new modules and code to it as really a community-developed platform. So it's a nice, cheap way to have access to analytical tools and ways to analyze genomes. So we welcome you to use it if you're interested. And in fact, all that data that I said we put up on Anvil, this is a nice success story. So we put one of our cases from our Center for Mendelian Genomics up there, and a colleague, Aaron Quinlan, who's a computational biologist out in the University of Utah, was testing out a new structural variant caller, and he used our data and in fact made a diagnosis of one of our cases using finding a structural variant upstream in the regulatory region of a gene, and we were able to diagnose this patient. So it just shows you the power of really enabling the sharing of the data and the sharing of tools and allowing our community to work jointly together to develop the best and greatest tools. I also have, over the last, I don't know how long it's been, 7 years or so, been working with the Global Alliance for Genomics and Health, which is a standards-building organization. In fact, I have a flight in a few hours to go to Barcelona for our plenary meeting there, starting tomorrow morning. And this organization is really, we've built all of these work streams that are shown on the left side that function on developing standards for large-scale genomics and cloud usage and genomic knowledge and so on and so forth. But we also define driver projects which are well-funded programs around the world working in different disease and biological areas that are defining the needs for data sharing and interoperability in their fields. They then funnel into our work streams that develop the solutions and the standards for that, which then go back to the driver projects for implementation, and there's a feedback loop really building what we need to enable a global community of data sharing and standards development so that we're really able to interoperate and compare a genome from one side of the world to the other. And so I would argue to the answer to this question, this bold prediction, in terms of everybody, it will be routine for any research laboratory, absolutely, but in part through really access to community-built platforms that we all are contributing to would be how I see that model evolving. There'll be democratization of putting those in your own pipelines, but really working to build them together. All right, the third one, which I'm going to tackle, is number 7, one of my favorite topics, and this is the one I actually had to speak on, which is the clinical relevance of all encountered genomic variants will be readily predictable, rendering the diagnostic designation variant of uncertain significance obsolete. Will we achieve this in now the next 8 years? So I'm going to give you a little data. Actually, this is data that's hot off the lab press, and that is so there's been this premise that insurance companies don't want to fund genome and exome sequencing because it generates all these VUSs, and that's a lot of uncertainty. It leads physicians to go do all sorts of tests, and it creates all sorts of costs. So we should not fund exomes and genomes. Let's do the panels. Then they won't generate all these VUSs. Well, my suspicion was that that was a wrong assumption. And so I thought, well, why don't we collect all the data from all the labs and answer the question? So I did that. I reached out to all the major sequencing labs across the US, actually North America. We have 6 kids in there from Toronto, and said, can you just give me the rates of inconclusive results due to VUSs and your diagnostic yield from all the tests you've run over the last 2 years. And that was a period of all of 2020 and 2021. So with all of these labs, we amassed 1.5 million tests worth of data. Now, 96.8% of that data was from panels, and about 3.2%, which is about 48,000 exomes and genomes, were the data we amassed. And if you look at just the simple side of this, the panels, 32.6% of panels were inconclusive due to a VUS versus 22.5% for genomic results. If you then compare the yield, positive rate, found the answer, panels were 10.3%, genomes and exomes 17.5%. So 2 observations. Genomes and exomes win on both arguments, and most of the results going out are actually inconclusive. This is a challenge for our entire clinical community also. This just gives it, by the way, if you didn't assume that, both those values were statistically significant, actually shown here. If you look at the distribution across the size of the panel, no surprise, the bigger the panel, the more VUSs that you create compared to the genome. And you also, if you're curious, in terms of the volume breakdown across panels, you'll see in these different bins in terms of what's being run. Okay, also we did look at the question is, well, why are panels generating more uncertainty, VUSs, than genomes? It's really related to how we report results. So in panel testing, most physicians don't give any phenotypic data, and the policy is really, if it's a diagnostic test, you report VUSs, likely path, and path variants, all VUSs. Whereas in a genome, it is true, you're generating thousands of variants you don't understand, but you can't put thousands of variants on a report. So what do? You ask the physician to give you detailed phenotype, and then you look at all your variants, and you only report those variants that are genes strongly correlated with phenotype and that have a reasonable amount of evidence to suspect causality. And that's why the genome results are actually much lower rates of uncertainty, because you're actually strategically thinking about which variants to put on a report. Also, in the genome side, it's well understood that trios are useful, where you can both phase variants as to what came from mom and dad, and you can detect de novo variation, which is much more suspicious for pathogenicity. And so if you look at the data from, in terms of diagnostic yield, trios have a higher yield for diagnostic yield. And also if you look at inconclusive rates, trios have a lower rate of inconclusivity. So good on all counts. But we never do trios in panel-based testing, just because it's like, well, I'll just do parental testing after I get the variants. But when you generate so many VUSs, you just can't follow them all up. Interestingly, there were 2 labs that broke their VUSs down into sub-tiers. VUS high, more suspicious to be pathogenic, VUS mid, and VUS low. Now, what you'll see here is that they're fairly distributed for the panel-based test, but on the genomic side, no VUS lows were ever even reported. And that's exactly what I'm saying. We don't report the variants that are not suspicious. We throw them out. and so you're only really reporting things that you're quite suspicious about. And so we really need to separate VUSs into these categories. And in fact, the new guidelines we are working on right now that will replace the 2015 guidelines will in fact divide into 3 sub-tiers and give guidance for how to classify into those tiers. So that's just some data around what's happening in our field today. Now, even if you can reduce the number of VUSs on reports by changing some of the practices we do, you still have VUSs. And in fact, if you look at ClinVar, where the laboratories are sharing their data, the largest fraction of variants, of unique variants, are in fact uncertain significance and some conflicting disagreement between labs. And that's a challenge because evidence is often unpublished, inaccessible, or there's simply no evidence. It requires a lot of time to interpret these variants. And so that has led me to be part of the ClinGen project, or Clinical Genome Resource. We're really bringing the community together to expertly curate information and define an authoritative resource for the community. And there are 3 grants. You'll actually hear from Sharon Plon later today, who's a PI of one of those grants. I am as well. There's actually a number of us that are PIs. And then there's also a number of smaller grants that are disease-focused now being funded. And there's a lot of things that ClinGen does, but the 2 key core curation activities are really in gene curation and variant curation. And so we have expert panels that are reviewing the evidence that's been published for whether there's sufficient evidence to implicate a gene in disease. And we've now, through these expert panels, curated 1,818 gene-disease relationships. Those go on our website, clinicalgenome.org. We also push them into a new database we launched called GenCC, where other groups are also contributing curations. And then once you've implicated a gene disease with sufficient evidence, then you can start curating the variants for pathogenicity. So we have other expert panels that are curating those variants, mostly focusing on the variants that are either conflicting between labs, differing opinions about it, and the experts can help resolve those, or in some cases, everybody agrees that it's a VUS, but there's multiple labs with data, and we can aggregate that data to better implicate the genes. And so we've got 2,000 expert panel volunteers from around the world, 45 different countries that are all contributing to this effort. And not only are the experts coming together, but a lot of those experts represent both clinical and research programs that are bringing their evidence to support the classifications. We have a large leadership team. There you'll hear from Sharon. Carlos used to be part of this group for a while, and you'll hear from him this afternoon as well. But there's about 100 staff within ClinGen that are doing a huge amount of work to make this whole machine work. Now, I didn't mention this, but when we do our gene disease validity curation, there's a lot of genes that have been published as disease genes in the literature. And in a lot of cases, there's not enough evidence for that. And they really shouldn't be put on panels, which the physicians assume are testing genes implicated in disease. And so we curate all these gene-disease relationships, and what we've found is that about 25% of the claims in the literature have insufficient evidence for a role in disease. So be careful what you read. Now, how are laboratories actually using this data? Well, this is an example. One of the most major changes with genes implicated in disease is actually around Brugada syndrome. There were 20 published genes, and the expert panel disputed 19 of the 20. And so when that paper came out and that data came out, this is Invitae's panel, they changed it. And they got rid of 19 of the genes. Actually, the way they work is the default panel is now at the top. If you just order the panel, that's what you get. But you have to, if you're a physician, you can manually add on the other genes that are in this preliminary evidence bucket. And that's how they've sort of catered their system to acknowledge the genes that have and haven't been sufficiently implicated in disease. Variant curation, we do hope to curate every gene-disease relationship across ClinGen. That won't happen on the variant side. We have probably billions of variants in our genome, already 2.4 million submitted to ClinVar. So we really focus strategies around developing standards and disseminating them, getting everyone to share their evidence in ClinVar so that you can transparently see what someone else has said and curated and build off of that and add to it. And then we focus on the interlab resolution and engaging experts in those areas that are really tricky. And that's sort of how we, as a community, work to deal with this incredible challenge of interpreting variation and its role in human disease. One of the biggest challenges, it's very labor intensive to get this evidence. It's like the experts come together, they're tackling a variant, like, does anybody have any data on this variant? And then somebody goes into their database, digs it up, types it into an email, and sends it off to the coordinator for that expert panel, who then downloads spreadsheets and emails and stuff. It's really not very scalable. So we really have to think about better scalable ways to share data and evidence so that we can scale the interpretation of variation. So one project we're working is really an extension of the gene matcher, or gene matching platform, the Matchmaker Exchange, trying to build a variant matching system. So if you find a variant, you could just enter it into a federated network, and what would get returned is, yes, we have a patient with that variant, and here's their detailed phenotype. Would be very useful. And in fact, some isolated systems have been launched, like MyGene2, Variant Matcher, Franklin Geno2MP. These are individual systems, and we're working on networking these through common APIs, like we did with Matchmaker Exchange, using standards built by the Global Alliance to help with the standardization of those APIs so that we can query across databases around the world to get more detailed phenotypic data on variants we're interested in. So this is a work in progress. We'll actually be talking about this in Barcelona tomorrow. So if we go back to this original bold prediction that there'll be no term VUS, well, I'm not so convinced that's going to happen. For a subset of genetic diseases, probably the most common ones, we will understand the variants that contribute to the largest fraction of disease. But I believe we will still have a long ways to go for the more rare diseases, and in fact, the rarest causes of all genetic disease, because even the most common diseases we deal with, cystic fibrosis, we still find novel variation all the time. It's a huge challenge. So I'm not sure we're going to quite achieve this bold prediction, but we will make progress. Okay, so the very last topic, let me just see how much time we have. We got plenty of time. So the last topic I want to touch on is that the regular use of genomic information will become mainstream in all clinical settings. And as many of you can appreciate, we use genetic testing a variety of ways today in medicine. We use diagnostic testing across the lifespan, where there are symptoms in a fetal ultrasound, symptoms in a baby, symptoms in adults. And then we apply the appropriate panel, or sometimes a genome and exome, to try and diagnose the cause of those symptoms. But we're increasingly using more screening or preventive genomics approaches, where we are testing asymptomatic or at-risk individuals. That can start at carrier screening or pre-implantation genetic diagnosis, NIPT, newborn screening, screening in adulthood for cancer or cardiac risk, or even use of drugs through pharmacogenomics. So lots of ways that we are and can use genetics in the context of medical care. So I ran a diagnostic lab at Mass General Brigham for 16 years. We went, from launching the technologies and sequencing every panel and all the way to whole genomes, offering them, and we still, despite being able to offer everything, many patients did not get these tests. And so I left that role about 4 years ago to become the Chief Genomics Officer at Mass General Hospital and formed a genomic medicine unit. And the goal there was really to work on the front lines, to engage the stakeholders in the medical arena, identify the pain points in implementing genomic medicine, and try to deliver creative solutions. And so we've done a lot of work in this space. We launched a preventive genomics clinic, really trying to tackle what we perceived as a gaping hole between primary care and specialty practice. most individuals don't get into specialty practice unless they actually have disease that relates to that specialty. But primary care is really not well situated to deal with at-risk individuals who aren't going to get those appointments in specialty care. So where does the woman who comes in and says, well, my mother died of breast cancer, my sister died of breast cancer, am I at risk? What should I do? And the primary care physician goes, I don't know. Let me phone a friend. Well, who do they phone? Where the cancer clinic is inundated with cancer patients. They actually can't see all these at-risk individuals. And so this preventive genomics clinic was started really to address this gap. And so that primary care could refer to us. We could help with, are you at risk? Should you have a test? Counseling, those sorts of things. Now, that said, we're talking about hundreds of patients a month that the cancer clinic can't see right now at MGH. We can't see them all in our preventive genomics clinic. So we also launched a genetics e-consult service. Here, the primary care or any physician can simply ask a question. Patient showed up with X, Y, and Z, family history of this. Should I offer testing? What test? And so we can just answer that question, develop templates, and then more thoroughly answer a lot of questions. And in fact, once we've answered the first question for a physician, that may then help them actually address that in their subsequent patients. So we're really learning and teaching through this process. It's a bit more scalable. We also recognize that there are some clinics that didn't have any genetic counselors. They really didn't know exactly how to order genetic tests or really apply them. And so we could deploy partial fractions of genetic counselors or genetic counseling assistants to help them where they needed it. And so we had this deployable genetic counseling service that's been very helpful. We also standardized consent forms across the whole hospital. And then, in an analysis of all the genetic testing companies being used, there were over 100 being used, which meant little integration with any of them in terms of the electronic health record and standardization of genetic results in the EHR. So we went through a process to interview all the stakeholders, define, look at all the tests and the labs, and define buckets of preferred labs, acceptable labs, and discouraged labs based on a variety of factors. And then that would prioritize those that we developed EHR integrations with based on the preferred category, which was a very small number. And that could help then really scale our use of genetics across the hospital. This is the team that has been doing this work, the core members on the side, but critical to this was actually having representation from every division and department across the hospital. So for each of these clinical areas, we defined a genomics lead for that clinic. And then they really help facilitate engagement with their department or division in the different areas of medical practice. And this has been really helpful at really addressing some of the pain points across the system. And one example, the kidney clinics weren't really using genetic testing at all. But when we came in and provided some genetic counseling and genetic counseling assistance to them, got them set up, ordering tests, immediately began to diagnose patients. In fact, Andrew Lundquist, who's the clinician lead for this clinic applying genetics, published a paper about just setting up this clinic and has found that about 54% of cases that he's been testing have had a diagnosis that was useful in determining the care of the patient. And I just put one example here of a patient who had a positive UMOD mutation that allowed them to put on ACE inhibitors and also then test the sister, who was a potential donor, but clear her for donation because she was negative for the familial mutation, which is important in that sort of treatment approach. So also in terms of our preventive genomics clinic, our goal here wasn't to really support the wealthy, curious people who want their genome sequenced. It's really to support this growing massive population of truly at-risk individuals who need genetic testing for a valid indication. And so we were curious to see who ended up in this clinic. About 58% were there because of a family history, and a smaller proportion were the sort of curious or concerned, but a lot of them for discussing both personal and family history. Also, nearly half were referred directly from primary care or the patients themselves. It was a self-referral requesting this clinic, and then about a quarter from specialists. And because most of these were targeted at-risk population, A lot of them did get testing, and 80% of patients met criteria and paid $0 for genetic testing because they actually met at-risk guidelines. So it's been quite successful, with one caveat, is just the number of patients we can see is still quite limited. And that's where we're really working on scaling a more genetic counseling only, for facilitating working with the specialty clinics to really scale the pre-test and post-test counseling aspects, but working directly with the clinics as opposed to our clinicians having to see every patient. And also, we've been tracking the e-consult requests that we've been doing, because I really believe that teaching the clinicians to be able to do this themselves is the only way we're going to scale. And so we actually tracked outcomes on the first 100 e-consults and what happened with those patients. For about 31 of them, there was a genetic evaluation, a referral that was recommended. But for another 6, non-genetics labs were recommended, or genetic testing was actually discouraged. The indication wasn't appropriate. Or we helped interpret previous genetic testing results or advise on a screening plan. Or in some cases, the bottom bucket actually said, you shouldn't be tested, your family member should be tested, and helping with that process. So in the end, I would argue that the critical elements to making clinical genomics part of everyday medicine is availability of affordable testing, an accurate genomic knowledge base, which will require sharing across our entire global ecosystem, a rich interface between research and clinical applications, global data sharing, access to genetics in the clinic, both in specialty clinics and primary care, access to preventive genomics at all stages in life. There's different things we want at different stages, access to genetic counseling services, and then integrating genomics into the electronic health record in usable, scalable ways. And with that, just to go back to this bold prediction, I do believe that we will have transitioned to genomes for most genetic services by 2030. But I'm not yet convinced that all specialties will have embraced genetics sufficiently by that time, and that we will have true equity in access to genomic medicine. We still have a long ways to go. And with that, I'd just like to thank many people across my team that do a lot of the hard work that I get to stand up here and talk about, so I couldn't do it without them. And hopefully we have a few minutes for questions from the audience. We're still on green, so we have time for questions. So I'll stop there. Thank you.
Thank you so much, Dr. Rem. As with our prior panels, please feel free to come to either of the 2 mics on the floor here. And if you're in the online audience, please feel free to enter questions into the YouTube live chat, and we'll relay them to Dr. Rem. So maybe while we're waiting for folks to come up, I'll just ask the first question here. I think you did an amazing job at highlighting the complexity and all of the organizational challenges that are required to steer us into the future, genomic variant interpretation. So I have to ask, I think there's a lot of buzz, of course, in our department and in this conference around the use of machine learning, artificial intelligence. Do you see this in the near future? So let's say, bold predictions by NHGRI in 2030. Do you see in 2030 this being a main tool that is in the arsenal of genetic counselors?
So I definitely think that machine learning and all forms of artificial intelligence are proving to actually be quite useful in a lot of different sort of areas. I think the biggest challenge, and one example is one really good tool for predicting the impact of splicing variants is actually the Splice AI tool that was trained on genetic data. And so that, I think those types of tools are proving their utility, but they're not the final answer, and we often need a lot of other evidence. And I think one of the challenges is, to train a tool, you need the primary evidence to train it on. And so if we don't have access to that evidence, then we can't train these tools. And so a lot of my emphasis on we have to share the data, and we have to make it accessible, queryable, is about the ability to use these kinds of approaches in scalable ways as opposed to, every time you see a patient, you send an email to me, gee, I see you submitted this variant to ClinVar and called it uncertain. Can you tell me more about the patient that you had? And then, we go and look up in the database, well, we saw it in a patient who was 2 years old, had a diagnosis, dilated cardiomyopathy. They also had 2 other variants, and they had these phenotypes, and that takes me like 20 minutes to go do that, and then they just read it, and it's used once, right? And no one else benefits from that exchange. It's all been private communication. So we really need to be able to have this data accessible at scalable ways if we're actually going to train these models and inform the answers. So yes. Go ahead.
Thank you. I'm Win Hyde at Beth Israel, Harvard Med School. Heidi, thank you for a stunning presentation. The question that came to mind was something that popped up in the 100,000 Genome Project in UK. And the question that was asked there, and I wondered what your answer would be, was what happens if somebody with a very rare genetic disease is known in the community to have that disease and is the only person in that city with that disease, or perhaps in the south of the country? How do you maintain the anonymity of that person when using Matchmaker, for instance?
Yeah, it's a great question, and this is something that we have tackled and actually published a paper on as guidance for submission to ClinVar. Because many of the laboratories, my own included, in fact, the data I had last time I looked, 67% of the variants we'd only seen in one person ever. And so you sort of ask, well, if I submit that information to ClinVar, am I somehow identifying that one person we found that variant in? And in fact, if you look across the entire ClinVar database, 75% of unique variants in ClinVar have been submitted by one laboratory. So, and you can look at our Nomad database in terms of unique variation across, 200,000 individuals, right? So there's an enormous amount of unique variation. So by sharing that unique variation, do we identify an individual? And I would argue only if you already know their genotype. So you have to know it to actually identify them by the genetic variation. With the exception of the patient themselves, they might figure out that, oh, that must be me in ClinVar. Now, they don't know for certain, because they may be the only person with that, but they don't know that there's another person on the other side of the planet. Now, your point was actually about the phenotype, right? And some of these rare diseases are, in fact, so rare that they have a unique constellation of phenotypic features. And so if you put detailed phenotype data in a database, you may be able to figure out that that person is that person, right? As you said. I think one of the things that we said in ClinVar is you can submit a variant, you can include phenotypic data, limit it to the features related to the disease. Don't say, and by the way, they also have, AIDS or something that is irrelevant and not good to share. So you're really limiting it to the phenotypic features. Again, can't prove that there's not another person with that set of features. That individual can find themselves, but we're not tagging their genome to them. And so you couldn't say, well, all right, there's the patient, they have all this, and gee, that's Sally down the street because I've seen her before, and now I'm going to look what other variant she has. They're not connected to the other variants in a public environment. Now, you could, if you share genomic data and you've registered in Ancestry.com and shared all your genotype data, and now you're starting to connect things, this can be done, and that's why people caution putting their names and their full genomic data sets from array-based testing online, and if you put your other genomic public data elsewhere. So, and long story short, yes, there are circumstances where you could potentially figure out that that must be Sally down the street, but usually Sally down the street would much prefer to have a diagnosis and understand why she has X disease than worry that people who already can recognize her phenotypic features discover that she's sitting in that rare disease Geno2 MP database that is actually, it's MyGene2 that's public. And we're not putting out in the public domain the full genomes. Those are sitting behind firewalls. They require controlled access where you define your use that we can monitor better. So I think thinking about the levels of access, the amount of data you're sharing, the detailed level of that data are all ways that we can sort of mitigate certain risks. Can we fully eliminate any risk? No, and that's where you have to balance what's the gain with what's the risk. And in most cases, or in all cases, allow that patient or that individual to make the decision themselves about sharing. So that's the best I can say on that. Yes.
Hi, my name is Luke. I'm a graduate student here, but I am representing the online audience. We got a question from Hyun Jung Kim from McLean asking, do preventative genomic clinics provide clinical services for young people at risk of psychotic disorders?
Good question. So this particular clinic that I mentioned is actually an adult genetics clinic. So that clinic is not providing pediatric support. But the general question in terms of psychiatric illness, so we are better starting to understand the genetic basis of psychiatric conditions. Some are more around as a complex trait with variation that contributes to risk, like schizophrenia, bipolar, other disorders that are less monogenic, where we would be looking for a single gene, single variant, or single gene at least, cause of that. So I would say, there is a brain disorders risk clinic at MGH. There may be at other locations that may deal with some of these. I will say that for a lot of psychiatric illness, the focus is actually on pharmacogenomics, because a lot of the ways that we use genetics for psychiatric illness is actually a lot of the medications are susceptible to metabolism by pharmacogenomic genes. And so that's probably in today's day and age, more psychiatric illness clinics are around the pharmacogenetic aspects than the diagnosis themselves, and with most of our understanding being limited to genetic contributions from GWAS studies and more complex genetics. But there are starting to develop some better understanding of some of the more significant contributors to disease. But yes, go ahead.
Hi, I'm Jane with Renal Fellow at Brigham. I'm asking in terms of the question of ensuring access to countries without that much money to pay for genomic tests.
Yes.
Representing Thailand now, they only covered BRCA1 and another colon-related cancer gene through a hard work of economic analysis that it would be good for the country. But then in order to integrate other variety of genetic tests, there's a ton of work that needs to be done that is done elsewhere to show cost effectiveness. And I wonder, from the big picture standpoint, are there resources that are building up to help these countries, including Thailand, to sort of implement genetic service in the low resource countries?
Yeah, I mean, I think there's some aspects of building the evidence base for determining clinical utility that are universal. Is knowing this information in advance going to change outcomes? Things like that, where studies anywhere in the world can contribute to this evidence base and are useful. But on the same hand, or on the other hand, in certain countries, there are very different healthcare systems, very different access to early stages of clinical workups and things like that. And so the practical aspects of how we think about implementation can be very different across different countries. And that's where, really being cognizant of the environment, the clinical infrastructure. Someone said, well, we should do newborn screening in, every country in the world. Well, what if there's not a system to then take those kids that have screened positive and work them up for a definitive diagnosis? You're just going to give back the genetic results and say, go have fun with it. You really have to have that infrastructure to, take the next step and do the diagnostic confirmation and then get them into an environment for workup based on those diagnoses. So I think we really have to think systematically about how to support all aspects of care, not just getting a diagnosis, but what do you do after that? How do you do cascade testing with family members and all of the things that come downstream? And that will require coordination, learning from each other, as well as recognizing unique aspects of every country and what they're dealing with. So I don't know if I completely answer your question, but try to tackle it, yeah.
So I think with that, we're out of time. Please join me in thanking Dr. Rem again. Fantastic, comprehensive keynote.